This is a video on how to image DNA origami grids using dSTORM imaging. The DNA origami grids we'll be looking at have 4 fluorescent dyes at each corner. First, let's clean the microscope slides and cover slips we'll be using for the DNA origami grids. Place the microscope slides and cover slips in their racks. This will ensure they stand upright during the cleaning process. Place this in a beaker and fill it up with 3 molar of potassium hydroxide. Next, we'll be placing these beakers in a sonicator. The sonicator uses ultrasonic vibrations to help remove any contaminants. The styrofoam shown in the video helps in keeping the beakers afloat. Leave the sonicator on for about 15 minutes. After about 15 minutes, turn off the sonicator and take the beakers out. Now, we're gonna rinse the slides and cover slips, but before that, we're gonna dispose the used KOH in our base waste. Next, we're going to rinse the slides and cover slips with molecule water as shown in the video. Repeat this process 3 more times. After, we'll repeat the same sonication process, but this time it's with ethanol. Lastly, we're going to dispose the used ethanol and rinse with molecule water 1 more time. Then we're going to fill up the beaker with molecule water. We will store the clean slides and cover slips this way. We're going to take one of the slides that will be used to image the DNA origami grids. Be careful not to touch the surface of the slides to avoid contamination. Next, we're going to spray the slides with compressed air, as air from the fume hood or other sources may contain grease or other contaminants. We'll repeat the same process with one of the cover slips. Now that we have a clean slide and cover slip, we're going to make a chamber for our DNA origami grids along with our imaging buffer. First, ensure the cover slip is large enough to cover the 2 holes drilled into the slide. One hole will serve as the entrance for flushing in liquid, while the other will act as the exit channel. To create this chamber, we're going to use a piece of parafilm. Cut the parafilm to the size of the cover slip. Then, carefully cut a large hole in the parafilm, leaving enough space around the edges to form a chamber. This chamber will allow us to contain the DNA origami grids and imaging buffer securely during our experiments. Remove the thin covering of the parafilm and place the parafilm on the slide, aligning the holes in the slide with the large hole in the parafilm. Then, place the cover slip on top of this parafilm. Next, place this on a hot plate to allow the parafilm to melt. This melting process will act as a glue, securely attaching the cover slip to the slide. Gently tap the cover slip with the pipette tip and check if the parafilm has turned more translucent. This indicates that the parafilm has successfully melted. Now, we're going to treat the chamber to allow the attachment of the DNA origami grids onto the cover slip. To begin, we'll introduce 10% polylysine into the chamber. Using a pipette, carefully dispense 70 microliters of the liquid into the entrance hole of the slide. Simultaneously, press a folded Kim wipe firmly onto the exit hole to draw the liquid through capillary action. Leave this to incubate for 30 minutes to an hour. Next, we're going to flush the excess polylysine with autoclave molecule water. Then, we'll be adding 20% of gold nanoparticle using the same process. After flushing with autoclaved milky water, we'll be adding the BSA biotin, and flushing with autoclave filtered PBS after. Next, we'll be adding the BSA. This time, we'll be flushing with autoclaved and filtered PBS. Now we're going to be doing the same process with streptavidin. Next, we'll be diluting our labeled DNA Origami grids to the order of picomolars with PBS and flushing it in. Shown here is the schematic of the DNA Origami grids with dyes at the four corners. Now, we're going to prepare our imaging buffer. We'll be using MEA, PCA, PCD, MgCl2, and autoclaved and filtered PBS. Given that PCD and PCA function as oxygen scavengers, we'll introduce PCD last to avoid premature activation of their mechanism. Due to PCD's susceptibility to temperature variations, we'll carefully add it to our imaging buffer inside a -20-degree freezer, as demonstrated in the video. After flushing in autoclave filtered PBS, then we'll be flushing in our imaging buffer. Next, we'll seal the open holes with another cover slip. It's not essential for this cover slip to be clean. Then, we'll secure the edges with nail polish to ensure a tight seal. Then leave this to incubate for 30 minutes. After preparing our sample, we're now ready to begin imaging. Our microscope setup, as shown in the video, requires a specific sequence for turning on our instruments to ensure proper functionality. First, we'll start by turning on the camera. Following that, we'll restart the computer to initialize all necessary software and connections. Next, we'll power on the AOTF and laser components to prepare for imaging. Finally, we'll activate the stage controller and ASI autofocus system to ensure precise control and accurate focusing during the imaging process. We'll be using a 60x all immersion objective during the DNA Origami imaging. Now, we're going to start the Micromanager software, which is an open software for image acquisition. To mount our sample, we begin by placing a couple of drops of immersion oil on the side of the cover slip attached to the parafilm. Then we carefully position this onto the holder and secure it onto the stage for imaging. dSTORM imaging requires a high-powered laser. This is controlled by the AODS controller in the computer. Next, click on Live on the Micromanager software to show a pop-up window of what the camera is observing. Then we're going to turn on the laser through the AODS software. In our case, since we're using Alexa 647 dyes in our origami, our laser used to excite it has a corresponding frequency of 53.64 MHz. Set the output to 2000. This can be clearly seen in the change in brightness of the light. After adjusting the Z focus, we're now ready to capture a movie of our dyes blinking. To set up our autofocus system in the Z direction, we follow a series of steps. First, we turn on the autofocus system by accessing the plug-in, device control, and selecting ASI crisp control. Once activated, we click on idle to initialize the system and then proceed to click on lock cal, waiting the calibration process to complete. Then, we click on dither, enabling the stage to make subtle movements up and down in the Z direction. Throughout this process, it's crucial to maintain the absolute error value of above 50. Once achieved, we click on set gain to establish the appropriate gain settings. We monitor how the error value changes relative to the 0 position value. Finally, to solidify our autofocus settings, we click lock, ensuring stability during imaging sessions. Now, we're going to select our region of interest where our laser is most focused. To do this, we hold down the Shift key and click and drag the mouse with the left click. Then we click on the leftmost icon under ROI in Micromanager. Clicking on the leftmost icon under Stage enables control of the X and Y plane of the stage. Now we're going to set exposure at 50 milliseconds and the gain set to approximately 100. Since we want to image an unexposed region of the sample, we turn off our laser and make slight adjustments to the stage position. Now we're going to click on Multi-D Acquisition. Here we can specify the number of frames we want to acquire. For DNA Origami imaging, we typically choose 10,000 frames, corresponding to about 15 minutes. We then select the destination for our files and start the acquisition process. Once started, we can turn on our laser to initiate the blinking event.
The movie acquired from Micromanager is in a TIFF format. Because there are too many frames to process, they're going to initially be split into two separate TIFF stacks. To do image processing, we need to stack them together to make one video. To do this, we're going to drag and drop the TIFF files to image J. Then we're going to click on Image, Stacks, Tools, and Concatenate. Since we turn on the laser after we start the acquisition, the first couple of frames will be dark. We need to cut those out. We have a plugin that can automatically delete the first couple of frames. To do this, we need to first install the plugin, go to Plugin, Macros, then Install. Then we have to click on the Delete Init Frames file. After we've successfully downloaded the plugin, we can go to Plugin again, then Macros, then click the Delete Init Frame plugin as shown in the video. To use this plugin, we need to go to the first frame and count how many dark frames we have. To be safe and not delete too many frames, we can plug in the number of frames slightly lower than the number of dark frames. Then we can manually delete the extra dark frames by going to Image, Stacks, and Delete Slice. Keep on deleting until your first frame is the brightest frame where all the dyes are on. Since we want to select the region where the laser is the most focused, meaning the brightest region, to ensure a relatively uniform laser power, we can hold down the Shift key and click and drag the selected region. Then go to Image, Duplicate, and follow the prompt. Once we have our cropped region, we're going to save this as a TIFF file. As you see, there are a couple of dyes that are constantly on. Those are the gold nanoparticles, and they only reflect light. We use these as fiducial markers to track the X and Y drift. We want to have a couple of these fiducial markers in our frame. To track these gold nanoparticles, we're going to select the region where only the gold nanoparticles are. To do this, we use the same duplication method as previously. And we're going to do this for at least 3 gold nanoparticles in different regions to capture translational and rotational movement. Then we save the TIFF file for each gold nanoparticle. To track the movement of the gold nanoparticle, we localize its position and track its movement throughout the video. To do the localization process, we use dSTORM, which is an open source localization software package. After opening dSTORM, we input the TIFF file of one of the gold nanoparticles. Then change the pixel size to 75.7 and the point spread function full width half maximum to 390. Then we're going to change the intensity threshold. We play around with this value to throw out any signals that are too small, which are not gold nanoparticles. These could be from residual signals of neighboring molecules. Then we're going to click on Add Output Module and select Display Progress, Image Display, and Localization Files. Then we're going to click Run. This image shown here shows the path of the gold nanoparticle. To check if our intensity threshold is too high or too low, we're going to open the localization file from dSTORM. The number of lines should be slightly lower than the number of frames of our gold nanoparticle TIFF file. If the number of lines is larger, we need to increase the intensity threshold from dSTORM. Now we're going to do this for the other gold nanoparticles. Next, we're going to localize the blinking dyes. We're going to do a similar process, but with the cropped TIFF file. But now we're going to set 350 as the full width half maximum, set 1000 as the intensity threshold, and have localization file and display progress as our output module. Now we're going to move on to our pre-written MATLAB code for drift correction and clustering algorithm. First, we're going to open the R1 file from MATLAB used for drift correction. Then we're going to run the codes in sections. For the first section, we're going to change the pixel size to 75.7 and then run the code by pressing Shift Enter. Then it's going to ask for the gold nanoparticle localization file. Then go to the next section and press Shift and the down arrow simultaneously. Then we're going to run the different sections in this MATLAB file as shown in the video. We're going to run the different sections to track the movement of the gold nanoparticles. Then the code will move the subsequent frames such that the gold nanoparticles will lay over each other. Do this for the other gold nanoparticle localization files. Then we're going to find the average drift correction from the gold nanoparticles. To apply this drift correction to the localization of the blinking dyes, we're going to run this section of the code and load said file. Then we run the other sections of the code, and we will end up with a drift-corrected localization file for the blinking dyes. To have a visual on how the drift correction works, we can apply the drift correction to one of our gold nanoparticles, as shown in the video.
This will output a drift-corrected localization file of the gold nanoparticle.
Then, to see this, we're going to go back to dSTORM, select Job, Replay, and then Minimal. Then we're going to select our drift-corrected nanoparticle localization file. Then under Output Module, we're going to select Image Display. As seen in the video here, now we have a stacked set of frames which shows no drift as compared to our previous stack. Now moving on to the R2 MATLAB file. In this section, we'll be taking out any dyes that are moving away from the frame after drift correcting it. First, we'll run the first section of the code, which will ask for the drift-corrected localization file of the blinking dyes. Next, we'll input the cropped TIFF file of the blinking dyes. Then we'll run the next sections. In here, We'll set the upper limit for the number of localizations as the number at the x-axis just a little bit beyond the end of the histogram decay. Then we'll run the next sections as shown in the video. This will output a refiltered dye coordinate file. Next, the R3 MATLAB file will output localization files for each dye. We do this by inputting the refiltered dye coordinates from R2 and the drift-corrected localization file of the blinking dyes. Now, in the R4 MATLAB file, it will filter out fluorophores that did not blink. In here we want to select our crop TIFF file of the blinking dyes, then the mean drift correction from R1 and the dye coordinates as shown in the video. Then we'll select the localization files for each dye from R3 and the refiltered dye coordinate from R2. Moving on to R5, we're going to compile all the good localization files into one file. We now have a localization file of the drift-corrected blinking dyes. To visualize this image reconstruction, we can use dSTORM similar to what we did previously with the gold nanoparticles. Once dSTORM is open, go to Job, Replay, then Minimal. Then we're going to input the localization file from R5, named Total Localization File. Then click on the image display under Output Module. Zooming into this reconstructed image, we're able to find a reconstructed image of the DNA Origami grids.
Looking at the schematic of the DNA grids, theoretically we're going to have 4 dyes within the diffraction limit, but due to issues with the dye labeling or blinking event, we're going to see some grids that have 1, 2, 3, or 4 dyes.